First part is sort of a mixture of personal reflections and meta-scientific sort of processes at a very, very broad level. And the second part is about the substance of research, which I've been doing over the last 10 years with the aid of many brilliant collaborators, none of whom have chosen to turn up to the talk, so I can accept all the plaudits, such as they are, for myself. So if I say I mean I did it. So I'm going to start with an anecdote, and it's from someone, it involves someone who's sadly departed only relatively recently, was a head of school at Cardiff, and he introduced me to the perils of middle management at university. And on one particular occasion where we were all sitting around in his rather palatial office, he had received a receipt, which many of us do from time to time, asking about what our research strategy is. Yeah, this is very important, especially to people who are at the upper echelons of the university. And he kind of mused that it's all very well developing such a thing, but he would often find it difficult to position exactly where he laid it when he wrote it. So he thought it was more difficult to find a memorable place for it than to actually generate such a motherhood and apple pie, as they sometimes call it, research strategy. And I think what he was alluding to was the idea that actually, as we all know as researchers, much of our lives are not so much driven by strategy as such, but as happy coincidences of one sort or another, either involving collaborators or involving the results of our carefully planned experiments with completely predictable results that happen to come out in the wrong direction. And those are the things that kind of grab your attention more and serve as a basis for future research. So so that's what I'm going to be talking about. This is not Father Christmas. Yeah, it looks a bit like a grumpy Father Christmas. That's a post-Christmas joke. Actually, as you all know, this is one of the founding fathers of experimental psychology, a guy called Pavlov. Now, Pavlov's research is, as you will also know, probably in greater detail than me, he's first started off doing basic kind of physiology of digestion. And this is a diagram. I don't think any of these things are to scale. That's the dog up there. That's the subject matter of the experiment. It's a bit like, you know, you have these fancy, we have something called Kubrick in Cardiff that has many machines that are not unlike this in terms of their complexity. And actually the real substance of the whole thing is the dog there and how its digestive system works. So he got the Nobel Prize, as you all know, for the work he did on digestion, quite rightly so. But he's probably in popular culture and maybe in experimental psychology more famously known for his work involving psychic secretions, as he first called them. So actually this is a lab, and a picture of a lab that he was given by the Soviet government. And over here, it says apparently it says experimental genetics of the higher nervous system. And over here it says observation and observation, presumably anticipating the multiple replication crises that we all go through from time to time, personally and as a field. So this is the kind of apparatus. It's not a sepia-coloured picture of the work that he was probably most famous for, is most famous for. And this is the stuff of serendipity, psychic secretions, and celebrity. See, a nice bit of alliteration there again. Fantastic. Love it. So this is a dog. It's my dog, if you put him on this table, wouldn't be standing still. Probably be jumping off. And this is the experimenter in his tie, doing experiments on psychic secretions where he would pair various stimuli with the delivery of food over here. This would come round. The dog would dutifully eat the food because he was a bit hungry. And you'd sort of see some sort of response, which was salivation coming through here, which sucked up into these bottles somehow, magically. Anyway, so it's probably, you know, if you and I were doing experiments on digestion and collecting as a measure salivation, and what we noticed was that whenever we walked into the room, before we'd even given the darn dog some food, it started salivating already. We'd think of multiple clever ways to get round that artifact, yeah? We'd think, oh God, it's terrible, I can't possibly do the study, so we do all sorts of things to get rid of that psychic secretion that happened in advance of food being placed in the animal's mouth. And of course, Pavlov was not like that. He identified immediately, I think, relatively immediately, the fact that these things were in and of themselves of interest. They weren't just corrupting his basic science. They were things that needed to be needed to be studied in their own right. And that's what he did for the remainder of his career, as you know. So this is a famous quote from another serendipitous scientist. Another alliteration, sorry about that, I didn't mean that one. So in the fields of observation, chance favours only the prepared mind. And Pavlos was clearly one such mind. So towards the end of his career, this is about 2 years before he died, he was obviously extolling the virtues of his latter career on the nature of psychic secretions. And he was talking about, you know, the various brain mechanisms and the alliances between association and learning in the brain, so to speak. And he said that psychologists seem to have also recognized the importance of his observations for our understanding of associative processes more broadly. So he was in no doubt of the significance of the results for our understanding of learning and memory. Now, I didn't generate this. Yeah, this came straight from the web. This is it called a Wordle? Something like that, isn't it? Is it Wordle? Something? What is it called? Word cloud in the shape of a brain. And it shows classical conditioning, or as we call it, Pavlovian conditioning at the center. And we have various things. So a central kind of experimental psychology here, associative learning, you've got all sorts of educational psychology down here, PTSD, all these things relating to his originally serendipitous finding. What I'm going to do today, I'm going to focus on 3 of the words, none of them rude, conditioned response, association, and this tiny one here that no one will be able to see from the back, but has probably had the most long-standing influence and still does on our kind of understanding of learning at a variety of levels of analysis. And that word there is prediction, okay? Prediction not in the sense of me making a prediction and it going wrong during an experiment, but rather prediction in the sense that that's what's being studied in Pavlovian conditioning. Or so we are told. Okay, so it doesn't look so good on this slide. Okay, never mind. I'm going to give you some conventional wisdom that incorporates those 3 important words here and give you some notation. I call it a conventional wisdom because it's mine. At the time, 10, 12 years ago. This is how I would describe it to students maybe. This is how it would be described when referenced to important models of learning and memory, including the Rescorla-Wagner model, famous model for its, it's being able to be applied across multiple domains of psychology. So there's a conditioned stimulus here, CS, and these can be auditory or visual stimuli that don't initially generate any responses of themselves, presumably. There's food here, that's called the unconditioned stimulus, and then this thing over here called the unconditioned response. And the general description of what goes on during conditioning, in which one stimulus is paired with another, is that the first stimulus comes to provoke a response that in some sense foreshadows and is related to the unconditioned response to the US. And that happens by dint of an association that's formed between these 2 memories. That's the idea, okay? And when I talk about forward conditioning here, I mean cases in which the stimulus comes on, the bell, it wasn't a bell, but anyway, let's call it a bell stops, and then the animal gets given its food. So that's forward conditioning, so where the bell predicts the occurrence of something that the animals like, food. Okay, now we're going to go into this is a personal journey. Sound quite right in this context, but anyway, this is a personal journey of 3 people who contributed to this work over the last 10 years. It's not all I've been doing, okay? I've been doing other stuff, but this is what I'm going to talk about today. And these 3 people are, so this is Sabrina Cohen-Hatton, who's 3 people in one, really. She was, when she arrived to do a PhD with me, and Simon Kilcross left immediately, of course, leaving me with the burden. Which was great, actually. Not that he left. Sabrina Cohen-Hatton, a firefighter, when she came into my office the few days after Christmas when she was due to start her PhD, she was like, I interviewed her, and when she came back several months later after finishing her Open University degree for which she got a first, she was very heavily pregnant, and her main worry was whether she'd be able to start on the 1st of January or whether we'd not let her start again. And I said, my goodness, yeah, anyway. So she was the person that won the BBSRC Innovator of the Year, and I was on her coattails, very much so. Eleonora Patitucci, I think it's pronounced, came and visited our lab from Bologna. Obviously, you want to come to Cardiff because of the weather and the food. And she eventually became a Cardiff PhD student in human neuroimaging. The fact that, that both of the other 3 people earned prizes of various sorts, and she went to do human neuroimaging is just a correlation. And she didn't want to come and work with me as a PhD student. Anyway, no, she's fantastic. Yeah, so she visited, and I'll tell you what prompted a lot of this research in a minute. And Ina Ilescu, who I'd met in this kind of fortuitous meeting at one of the holidays in one of the visiting professorships in Granada. She later became a Cardiff PhD student because the University of Sydney, thank God, refused her a position. So she came and worked with me, so fantastic. Anyway, she went on to win one of these prestigious prizes for her PhD research, which I'll be talking about in a bit. Yeah, so initial meeting with Eleonora was really interesting. So she came to my office, it was very early 2015, sat down and started talking about what she wanted to do in my lab. Right, okay, fine. You've been invited over. And she was talking about something that Sabrina had done for a PhD. And those that know what Pavlovian to instrumental transfer means know, and those that don't need to know, if you see what I mean. So she was interested in what Sabrina had done for a PhD. And she was saying, yeah, well, what I want to do with that procedure that you developed, it's not our procedure, but that we replicated in a variety of ways, is to understand something that she said, the individual differences in the nature of conditioned responding. She didn't quite put it like this. And I said, so I don't care about the Pavlovian to instrumental transfer so much. What are you talking about? Individual differences in the nature of conditioned responding. What do you mean? And she said, well, it's kind of well established, 3 or 4 years ago, at that time, that when animals receive simple insertions of this lever into an operant chamber, here pictured, and that lever withdraws and then is followed by the delivery of something like food or sucrose, the animals are known to show 2 sorts of behaviors. One of them is called sign tracking, where they kind of look at the sign and go over and play with it. They don't have to get food. It's a Pavlovian response rather than an instrumental response. So they go over and play with the lever, and they also put their head in anticipation, presumably of food, in the food well. But she said, well, what do you mean? They just randomly do this. I mean, you're talking about individual differences. What do you mean? And she said, well, no, some of them show extreme sign tracking. They show what they've learned in terms of one sort of response, and others show what they've learned in terms of this other goal tracking response. Really? This very simple procedure, simple Pavlovian conditioning procedure, gives rise to these rather extreme, I mean, it's shades of grey. I mean, some of them are really extreme sign trackers, some are really extreme goal trackers. But there are these systematic individual differences that, apparently, that are, consistent across days of training. And that sort of piqued my interest. I was slightly dubious, I have to say. So the kind of technical way of putting why it might be interesting at all is that, Pavlovian conditioning generates behaviors now that don't just reflect the nature of the US, the fact that food's delivered in this funny little food hole, but also the nature of the stimulus that predicts the food. They go over and play with the lever, or into it and engage with it in ways that can be measured automatically. And there are individual differences in the extent to which they exhibit what they've learned in terms of these 2 behaviors. Now, as everyone will not acknowledge who knows a bit about, even a little bit about Pavlovian conditioning, is that we know that the unconditioned response to a conditioned stimulus is not identical to the response that you'd show to food itself. It kind of resembles it. But there's also this response to a guy called Peter Holland and Bill Timberlake in the 70s has shown that the nature of the conditioned response can also reflect the nature of the conditioned stimulus rather than the unconditioned stimulus. Now that's poorly understood why that should be the case, given the conventional wisdom that I set you off with, or that I had at the time. But the other idea that there are these kind of very systematic individual differences in the form of the response that animals express their learning in seemed really an anathema, at least to a general process person like myself. So rather than just saying, well, we're not doing that, I thought, oh, that sounds really interesting. And we were at the end of a BBSRC grant, and we had a little bit of cash left over when they allowed you to keep such things and put them in strategic holding accounts, and we don't have such things anymore. Anyway, so what we did was we kind of set out to replicate in using experiments that were a bit more tightly controlled than those that had been published by Shelley Flagel, either in Nature or in Neuropharmacology. We set out to replicate those results, and our minds were truly sort of unprepared about what the real systematic importance of those individual differences might be. So here's the first experiment. How am I doing? Blathering on. I'm all right for the moment. So the experiments modelled on those of Shelley Flagel, and what happened was that the animal would sit in this. They'd be hungry. They'd be deprived to 85% of their free feeding weight, their after Christmas weight, and what they'd get is so they'd be deprived to 85% of that. And then they go into a little Skinner box, and they get a lever would come out and be followed by food, and that's all that would happen. And we had 32 of the little fellows doing this, and we just monitored their tendency to put their heads in the food well or go and play with the lever, which you can do both of those in an automated fashion. You don't have to video record them and score their behaviours. These things are automatically measured by the boxes, so low cost to me anyway. So there's a proper picture of something that resembles our Skinner boxes there, and we're actually giving the animals two levers. They don't ever come out at the same time, as pictured here. You get one lever that's followed by sucrose, and you get another lever that's just a dummy lever that's not followed by sucrose rather. And the results are fairly amazing. So what you do. Sorry, this is a bit complicated. What you do is at the end, say at the end of training over here. Yeah, on block 10, you do a median split. You divide them into animals that do a lot of sign tracking and animals that do a lot of goal tracking, and you call them the sign and goal tracking group. And then you plot their discrimination between the lever that's followed by sucrose and the lever that's not followed by sucrose as it emerges over the trials. Yeah. So what you can see there is that for the sign tracking group, they are most likely to show that discrimination learning in terms of their lever presses or their sign tracking, going and playing with the lever, and are less and don't show it so much in terms of their goal tracking. This is just a median split. This is not taking the extremes on the distribution. Whereas in the goal tracking group, they show the reverse. So the animals are showing their discrimination, their learning in terms of food well activity, and rather less so or if anything sometimes, in terms of their lever pressing. So even those observations replicate what Shelley had done, and are kind of interesting. So we wondered, as general process learning theorists, about what might be the origin of those differences in behaviours, and we thought, well, perhaps. I mean, people allude to differences in mnemonic structures of the form. The familiar ones in animal learning are stimulus-response associations and stimulus-outcome associations, so stimulus-goal associations and things that are kind of habits, popularised in the context of instrumental conditioning by Tony Dickinson. So we thought, well, okay, perhaps the animals, this would be interesting in and of its own right. Perhaps some of the animals tend to be more habitual, and one of the responses reflects that, and some of the other animals maybe are more goal-directed, which would be interesting, and their behaviour is reflected in the other response. And we weren't particularly concerned about which response that might be. So the traditional way of assessing those sorts of ideas is to manipulate the value of the goal in some way, or the outcome, as it says here, in some way. So what we did was we did another experiment. This is a second experiment. And what we did, we had two levers coming out at different points. One of them was followed by food, and the other one was followed by sucrose. And what we assumed would happen under those circumstances is that the animals would develop these individual differences in sign and goal tracking. So a sign tracker on the left lever, sign tracker on the right lever, but just for different outcomes. And then what we'd be able to do was to manipulate the value of the goal to see if it influenced one response rather than another response, okay? Yeah, we didn't care which response it was particularly, although we had a sneaking feeling it would be goal tracking rather than sign tracking, whereas Shelley-Flagel's ideas assumed the opposite. But anyway, that doesn't matter for the moment. So what we're going to go on to do was give the rats lots of either sucrose or food, and then see whether that satiation immediately affected one response or another to the lever on which the outcome had been devalued. Now, so you can forget about these two bits actually, because we didn't get that far, because our original assumption about a goal tracker or a sign tracker being a sign tracker on each lever turned out to be wrong. So a serendipitous observation was that, so if we look at this figure now, and this is a more conventional way of presenting individual differences. So this figure here shows responses on the left lever and responses on that lever again, but on a different block. So this is on block 5 of training, this is on block 6. Scores that are negative are sign tracking, scores that are positive are goal tracking using this measure. And what you can see is that if you look across successive days, there's this rather lovely correlation between their bias to show sign tracking or goal tracking on block 5 and block 6. And you can do the same with the other lever. That's also fine. Don't forget, I haven't done any satiation because of the results that are in the bottom half of the figure. Same is true of the right lever, irrespective of whether it's food or sucrose, actually, that's counterbalanced. They show a nice correlation between what they did in one session and what they did on the next session, sign tracking, goal tracking. The oddity was that when you looked across the levers, even within a session, the animals that were sign tracking on one lever were not necessarily, they weren't sign tracking on the other lever. So there was no relationship across those two levers, which meant it was very difficult to go on to do what we had intended to do. So that's a weird serendipitous finding. And what it suggests is, unlike what I think what we call the sign tracking mafia, and they probably call us the goal tracking mafia, but anyway, sorry, that's naughty. So what Shelley-Flagel might say and is that, okay, so if you're a sign tracker, that's because the sign, the lever, is attracting some incentive salience, and that should be the case irrespective of whether a lever is paired with sucrose or food. So they should show the same bias on the two levers, and that's not upheld here. You're not a sign tracker per se. So the results seem to suggest that it's something to do with the outcome that generates these two different behaviours on an individual basis, presumably, or it's something to do with the left or right lever, or it's some interaction between those two. And we'll return to that idea later on in a more kind of theoretically oriented way. I should say as an aside, we went on to do other experiments where the two levers were paired with the same outcome, and then they did correlate. The tendency to sign track on one lever, goal track, correlated perfectly well with the others. And we also went on to show that actually when you do the experiment slightly differently and you sate the animals on food, then it affects goal tracking more than sign tracking. So we did that, but that went on in the background. And Ina then went on as a PhD student to do a lot more kind of exploratory work on what turned out to be something that would guide our theorizing. So these are the sort of questions that, at least in animal learning theory, guide our kind of understanding of, or at least the answers to them should guide our understanding of what's going on. And one is, you can ask these three questions in black about anything that any animal learns or any person learns. So what's learnt? What are the mnemonic structures or associative structures, in our case, that underlie the change in behaviour you see? What are the learning rules involved in those changes in behaviour? And how's the learning that you assume to be going on in the animal's head, presumably brain, how's that translated into behaviour? So there's got to be some translation, performance rules as well. So this is the Riscula-Wagner model. So you've probably never heard of it, a very recent model. 1972, and it's dominated the field and other fields for many years. And it adopts, I mean, it doesn't quite say what I remembered it to say when you go back and look at it closely. It actually has very little to say about any of those three questions. The one thing it's specific about is the learning rule. Everything else is implied in other people's minds. They get different interpretations of it because they certainly don't say what I thought they said, or I, you know, over the years had kind of gradually come to a position on. But anyway, so imagine they said this, that what I said to start with was true, that during Pavlovian conditioning, the memories of these two things the link between the memories of these things are strengthened. They're strengthened according to this rule here. The rule is an error-correcting rule. It's one that is, there's a prediction error here. So the change in associative strength on a given trial between this and this is dependent on the difference between the maximum associative strength that a US can support and the strengths of all of those stimuli that are present on that trial, with that difference being multiplied by the saliences or the intensities of the CS and US. And that's maybe pretty much what's dominated the field for 50 odd years. That idea that it's, you can see where are we coming from there? What do they say about performance? So I'm just try and persuade you that they say very little about anything at all, in fact. So Rescorla-Wagner, in providing some mapping of associative strength into behavior, they say it's just sufficient to say, well, the magnitude or probability of a given behavior preserves the ordering of the associative strength. So if the prediction is that this group has higher associative strength than this group, there's going to be a difference in that direction, basically. McIntosh, another very recent theory, suggested that it's sufficient, and he used the same word, for present purposes to assume that the associative strength of a given stimulus has a monotonic relationship to behavior. Yeah? And Pierce Hall say, we're not going to talk about that at all, actually. They had other pressing issues to consider. Yeah, so those theories are remarkably, all of them, are remarkably poor explaining the fact that we all knew about, that behaviors generated by Pavlovian conditioning reflect both the nature of the US, as Pavlov thought, but also the nature of the CS, as he also knew. And they're also remarkably difficult for them anyway to explain why there will be individual differences in how associative strength is mapped onto behavior. So you can see how ludicrous the idea is, the monotonic relationship between associative strength and behavior, when I ask the simple question. This is about the initial experiment that Eleonora ran. So which group of these two has the highest associative strength? Which one of those two groups? And you say, well, if I'd only measured sign tracking, if I'd only measured sign tracking, then the sign tracking group would be better than the goal tracking group because they show better discrimination, yeah? In contrast, if I'd only, and some people do only measure one of these things, if I'd only measured goal tracking, then I'd say the goal tracking groups as better learners, they've got better associative strengths than the sign tracking group. So the mappings just don't make any sense in terms of those theories of associative learning. So I'm not sure whether I ever put it this explicitly, but it seemed to me that this is what we were thinking at the time. So what we needed was some sort of general process model of learning that allowed the conditioned responding the nature of it would be influenced by the nature of the CS and the unconditioned stimulus, and that there could be individual differences in these behaviors. And what we also, in answering those two kind of pressing concerns, what we ended up doing was generating a model that could explain a huge variety of things that had been taken either to be inconsistent with an associative theory or had prompted people to start thinking, well, you have to include things like attention in these models. It can't just be association. You have to have an attentional parameter that can change as a result of experience. Okay, so I'll come back to those in a bit. So although it looked very clever when we presented it in the two large papers, that we were able to explain all this stuff, but we were able to explain all this stuff for free. The model was generated to explain a relatively limited data set. Okay, so the idea that we came up with, but Azratian had done many years before, was that the associative structure looked more like this. And this is my rendering of his figure. It's a faithful rendering of his figure. So we have two stimuli coming on. You might call that your stimulus 1, stimulus 2, or your conditioned stimulus followed by your unconditioned stimulus. And what he reasoned was that the memories of these two things, with these micro features within them, that can each become connected to one another as well, but these two nodes can become linked to one another by a direct conditioned connection, which he said, so a standard predictive, if you will, link, but also that the unconditioned stimulus can become linked by a reciprocal conditioned connection to the conditioned stimulus in terms that I've been talking about already. And one of the bases for that associative structure was that, as Pavlov had kind of anecdotally noted before, the response to the conditioned stimulus didn't just reflect the response to the unconditioned stimulus, but also some of Azratin's colleagues did experiments where after the animals had received conditioning, they would present the animal with the second stimulus for the first time, but just presented on its own, and it would generate responses that reflected the nature of the first stimulus. So if you presented food, you'd get responses that not only reflected the fact that food was there, but also generated orienting responses of the form that the first stimulus had been observed to elicit itself. So that was his evidence that so the reciprocal connection provides some grounds for generating behaviors on the basis of the conditioning procedure that reflect the nature of the conditioned stimulus. It won't, in and of itself, generate extreme individual differences because, of course, even if this connection is very strong, its impact upon presentation of the first stimulus is going to be still going to be relatively weak. It's going to be dependent on the strength of that association. So, for example, if that's 0.5 in terms of its strength, and this is 1, which is the maximum, that will still only mean that this gets generated 0.5, and that is generated 0.5. You won't get more responding on that basis. So there must be something else going on. This is the stolen version of Azratin's ideas, in which we have a condition in the untrained animal. We have an unconditioned state in which the CS and the US can activate various different sorts of responses that are different depending on what those stimuli are, and that as a result of conditioning, reciprocal independent links are formed between the conditioned stimulus and the unconditioned stimulus. We also steal, so we haven't only stolen Azratin's idea without asking him, we also steal the Rescorla-Wagner model for the strengthening of those two associations. We simplify it slightly. We reduce the number of free parameters, which looks like 3 in the Rescorla-Wagner model, but is in fact 4. We reduce the number of free parameters to 2, the perceived intensity of the CS and the perceived intensity of the US. That's just a constant to balance the sides of the equation. So not psychological science, but psychological review, even better. Yeah, so the reason it's called Heidi, I spent longer developing the name for it than I did stealing the ideas, borrowing the ideas. And originally, there's not anyone old enough in the audience to have read Heidi in its no. So it's 1985. 1985. I might be very close. I feel it sometimes, that's for sure. So in 1985, there wasn't just a single book called Heidi. It was separated into two parts. One of them was called Her Years of Wandering and Learning, and the second bit was how she used or expressed what she'd learned. That's why it's called Heidi. It's not because the letters have me as the first letter and Dominic as the second and Ina as the third. It's not anything to do with that. No. And it's definitely not how excitation and inhibition determine idiomotion. It's definitely not that. Okay. So we've dealt with the associative structure, which I'll come on to in a minute, provides a way of explaining a whole raft of other effects that have been explained in more complex terms by other models. It's this bit that does this is the performance rule. So if you think about sign tracking, what we say is that, okay, so you combine the associative strength in some relatively trivial way of the forward and the backward association, and then you distribute their impact in terms of the relative perceived intensities of the CS and the retrieved US, because you're presenting them with the CS, so it can't be the US's actual value, it has to be its retrieved value, which is determined by its associative strength. And the extent to which you do goal tracking is simply determined by the reciprocal of that rule. So that's what's left over, if you like. So it's a slightly odd idea. Okay, so you've got the reciprocal associations by stealing them from Azratian, and then you've got this rule that now requires the animal in some sense, or the model at least, to compare these two very different things. You know, the perceived intensity of a stimulus like a lever or a light, and the perceived intensity of food. That's a really weird thing for someone to suggest that they can do. But I'll just show you first that it works. So if you run simulations, these are very rudimentary ones done with Excel. Someone managed to attract another person who does more than Excel simulations to do the proper stuff. But this was my half-baked way of doing it. So what you do is you look at the distribution of associative strength into either sign tracking or goal tracking, and all you're doing is following through on setting your alpha for your conditioned stimulus to lower or higher than the value of your unconditioned stimulus. And under those circumstances, what you can see is that the distribution into behavior in terms of performance tracks the largest of those alpha values. So it all does work, but whether it's plausible or not is a completely different matter. However, one reason to think it has flaws is that Nick McIntosh, God bless him, this is a paper published in Nature. This is the figure from the Nature paper. Yeah, it's done with a rotring pen. Yeah. I've still got mine. It's running out of ink. Times was hard then as well. So what he did was a really clever experiment. So with Over, he got the animals to learn an intensity discrimination where a bright light was followed by food and a dimmer version of the same light was followed by no food. Did it the other way around as well. Or he did the reverse discrimination where it was the dim light that was followed by food and the bright light that wasn't. And then he transferred them to a different dimension, another dimension which was now an auditory cue, and the designation of the auditory cue as loud or quiet either mapped onto the loud or quiet light or didn't. And what you can see here, sort of, take my word for it if you don't want to bother to encode it, when you get transfer between an intensity discrimination where it's the loud thing or the bright thing that's followed by food, it generalizes well across these modalities, and the reverse is also true. If you were doing it on the basis of the dim light being followed by food and the bright light not, then you get good transfer to the dim noise, the quiet noise, and the loud noise, and less generalization to the others. So at least within those, within the bounds of those sorts of stimuli, you seem to have some basis for saying, well, animals can compare across vastly different stimuli the relative intensities of things. Okay, so now all you need to do is to say, okay, so we've got a general process model that gives you conditioned responses that reflect both the CS and the US, sign tracking and goal tracking. All we need to say really now is, well, for the animals, sleight of hand now, all we need to say is that for the animals that are sign trackers, they, for some reason, find the light more intense than the food, and the reverse is the case for goal trackers. Well, you've already seen the simulations. When I mess around with the alpha values of the conditioned stimulus and the unconditioned stimulus, you see these crossovers between how learning is exhibited. Okay, so that's all you need to say. If anyone's thinking, why is that the case though? That's a good question. I might not be able to answer. Okay, so this is a personal journey, so is it taking too long? Oh yeah. It's a personal journey. Double-decker bus, another psych review, not psych science paper. Within 2 years, this is I think they call a bog off. This is buy one, get one free. So we applied the same logic to all manner of other things that are relatively important, at least for our field, that extend the range of conditions under which conditioning can have a repercussive effect on other stimuli associated with stimuli that are associated with food, called higher order conditioning. This, alongside the original model, was able to explain a vast array of things. So these things, you don't need to know what they are. Downshift unblocking and latent inhibition don't need attentional factors. unequal change in associative strength when two stimuli with different associative histories of conditioning compound. Rescorla's brilliant research from the noughties, which undermined his own pooled error term, you could now explain in terms of the model that we developed, higher order conditioning, and then the timing of responses we can also do, which I'll come to later, and why the intensities of the CS and US impact not only the rate at which asymptote is reached, but also the asymptotes themselves. So this is an experiment by Imada, and what it shows simply is that if you train an animal on a conditioned suppression procedure in which the shock is always the same, but the conditioned stimulus varies between 0, in other words not there, and very bright, then the asymptote that's reached isn't the same in those groups. In terms of the Rescorla-Wagner model, the asymptote should be the same, and actually in terms of our model, the asymptote should reflect both the intensity of the CS and the intensity of the US. This is not explained well by any of the models that I previously mentioned, those old models. This is showing that our model does, when you're looking at associative strengths now, it does the business. This is what you get from the Rescorla-Wagner model. For the forward association between the CS and the US, they get to the same asymptote, but at different rates. This one faster than this one. This is the backward association being stronger when the CS has more association. And this is their combined value that we use to distribute into the different forms of responding. Okay, now, Ina left. She went for the Ministry of Justice because she'd have a more, the work-life balance thing loomed for her. But at the same time, we managed to attract this guy who's fantastic and hasn't got a job. Employ him. So he has an engineering and psychology background, which meant he could not only fix our aging Skinner boxes, but he could also do the sums properly and design experiments, and he was a fairly good neuroscientist as well. Absolutely fabulous. And I don't know why we didn't employ. Nothing. So, I mean, one of the obvious predictions that come out of the whole thing is that, okay, so if you're saying that during forward conditioning, where a CS is followed by a US, you get associations running in both directions, what about if I did it the other way around? What about if I presented the food first and then followed a bit by the conditioned stimulus? That surely should also lead to, other things being equal, reciprocal associations between the two. And this is a well-used a quotation from Nick Mackintosh's Bible. Nick Mackintosh's Bible says, all in all, there is little reason to accept the reality of backward conditioning. In other words, this prediction is false. And actually this quotation and the empirical, the evidence that he used to support it is one facet, one strand of evidence that's been used to kind of emphasize the idea that in conditioning and in other forms of learning, prediction and prediction error are what is key in everything. And if you don't get backward conditioning, it's obvious why you don't, because the CS is not, it's not predicting anything, is it? There's nothing there. Tony Dickinson, however, always says something smart. So he said, well, it's a bit odd. So if I'm standing with my back to someone and they come and stab me in the back and I look round, sorry, this is a joke from Greg Higgin, no one's laughing. So and it does sometimes happen, people stab you in the back and you look around. You might want to avoid that person in the future in spite of the fact that there's no prediction between that person because it's happened in the wrong order. Yeah? Clever man. So this is to make it more concrete. So this is another sort of, it's just post-Christmas sort of thing. So this is Dr. Dolittle, yeah? So this is a push me, pull you. Yeah, you know about push me, pull yous. You know all about it. There's a description of what they are here. They had no tail. Well, of course they don't. And they had sharp horns, and there's a bit here that I've left out because not very delicate framing of things, this guy, So I took that bit out. Because no matter which way he comes toward you, you can always see you. But actually it's quite funny because in that picture, what you can see is that this one experiences Richard Attenborough, naughty man, poking him in the tummy, we'll call it there. This one experiences the tummy poke first and then turns to see him. It's like he's being stabbed in the back, yeah? So this is the basis for the experiments that the Victor ran. And so the basic question was, well, does backward conditioning produce changes in conditioned responding in a way that Mackintosh thought they really didn't? And evidence since has been very sparse and not very convincing, I have to say. So the experiments are, they look like this. So there is a series of experiments. This one is a simple one. They have 2 levers, not one. 2 levers, 1 lever is followed by food and 1 lever is preceded by food. We monitor goal tracking and sign tracking, and then we give them probe trials in which food is omitted because you don't want food to be influencing their ongoing behaviours. And you do that over many sessions, and this is just pooled over those many sessions. And what you can see here is that both forward conditioning and backward conditioning produce substantial changes in goal tracking. But had we only measured sign tracking, we would have been consistent with McIntosh's conclusions that it really doesn't, backward conditioning doesn't really work. And it's not just that the 2 procedures sort of a bit like predictive learning, that they're essentially the same sort of procedure. Because over here, as a sort of secondary observation, again, it was based on Ina's work actually, but that itself was, we were recording across the lever, so 10-second lever, in 2-second epochs what was going on. And what you can see is what Pavlov observed, that over the course of forward conditioning, the animal gradually ramps up its goal tracking, yeah, as if it's anticipating more and more that it's going to get food. It's timing, yeah. And in backward conditioning, the reverse is the case. So at those initial segments, bearing in mind that the food isn't presented on these trials, the animal's sort of coding where food, they've learnt that it's the beginning part, if you will, of the conditioned stimulus that is most important to them. So backward and forward conditioning backward conditioning not only results in substantial conditioning in the way that we would have hoped, but it also results in this kind of timing-like behaviour. It's not timing, actually, we don't think. What we think is happening is that the rats code where food's going to be delivered, and they do it by dint of if you imagine a stimulus coming on, the first part of it is going to be relatively intense. It pops out of the blue, a lever comes in, very salient, and then over the course of the stimulus, there's this short-term adaptation or short-term habituation process that means that the last part is less intense than the first, which provides a basis for them discriminating the first part from the last, a bit like over in McIntosh's experiments, but here you're not directly manipulating the intensity of the stimuli, you're relying on a sort of fading trace, even though the stimulus is there. So short-term habituation gives you a difference in intensity which the animals latch onto in order to show these timing-like behaviours. It's not timing, we don't think. There's some stuff on the brain. I don't know anything about the brain. I don't know why I'm doing this. Someone's going to ask a question about exactly this bit. Okay, so in Shelley's original experiments, what she did was she looked at these phasic dopamine responses in nucleus accumbens core, and what she found was in goal trackers, that response across the epochs of the lever, the duration of the lever went up, and she also observed that in sign trackers, so if you look at this one, it initially went up and then went down. So these patterns are exactly match. They're similar to the ones that she observed in her goal trackers and sign trackers. These are all the same bunch of animals rather than being split into 2. Okay, so humans, you're all here. So when you run out of money with rats, when you've got time on your hands, you go off and do impacty work with the emergency services, and you also do some experiments in people. These are experiments trying to look at the same sort of deal in people, forward and backward conditioning. And having read Mike Kahana's very fine papers on this very issue, we decided on this design. So in the middle of the screen, there are coloured patches coming up. They don't have these labels, A, B, C, D, on them. So this is just to help you understand, help me explain the experiment to you. So they get these are all intermixed in a random sort of order. They get yellow followed by green, green followed by blue, blue followed by red, and red followed by yellow. Now, what the situation in human memory, at least as I understand it, and I'm no expert, is that, at least as expressed by Mike Kahana, he thought that actually when you controlled for various things, that actually the associations between, for example, these 2 patches would be bidirectional, but it would be just one association that went in both directions. And of course, one association cannot have different strengths going in different directions. So that was his view, at least it was 20 years ago. I haven't kept up with him, he does too much. And after several blocks of training like this, they get tests in which a yellow patch will be presented and the animals, they are animals, our students can be animal. Everyone's students can be animal. They're asked what, out of an array, it wasn't just this simple, it was an array of different colours over here, which one went with the yellow, basically. And of course, if you only form forward associations, then you'll always say green. If you form identical association between A and B that can work in both directions and between D and A that can work in both directions, you should be equally likely to say green as to say red, yeah? And C is just our control stimulus in a way. And we show through simulations that a dumb Rescorla-Wagner model and a dumb completely symmetrical model doesn't predict any differences between whether you say green after yellow or whether you say red. In the case of having these dual associations that can have different strengths, you could get that difference. So you could get the forward one producing, I'm going to say green, and to a certain extent get the backwards one saying you get red. And we did a number of experiments like this because we could, and it's cheap. Varying things that are important. And what you can see in each case is far from being symmetrical. The people they get the forward association. These are probe tests done after different amounts of different blocks of training. You get the same basic pattern, which is the forward association is much stronger, or at least as expressed here, than the backward one. But there is still some tendency for the students, as they are, 30 students, 30 students, 60 students, 30 students, roughly, and it's all within subjects. And the error bars are on there, believe it or not. Yes, the blurry bits. So in each case, you can see that there's this tendency for them not only to act on the forward association, but to some extent say that the reverse also works. So to use a backward association, and that must mean that both associations are formed rather than being a single one that works in both directions or one that only works in one direction. And so why does that happen? Well, the obvious reason harks back to something I said earlier, which is if you imagine that the perceived intensity of a stimulus declines over its presentation, and that the intensity at that point determines the backward association and the forward association, well, you're going to get a higher forward association than backward association. The backward association is based on a lower perceived intensity than the forward association. Forward association is based on the intensity at that point. That's the idea. That's why you can get different strengths of their forward and backward associations. This is my foray into neuroscience again. God, so what recently Catlou et al. Do in this wonderful paper in Nature Neuroscience, so it might be true, might not be true, was just look at this dopamine response just to a stimulus. There's no reward involved, there's no prediction error, you just present the stimulus, and the next thing you do is you present the stimulus again. And what they observed was that this phasic dopamine response that I was talking about before looks like it declines over the course of a stimulus, whether it's a light or a tone or anything. So there's some evidence that the story about dopamine might not all be as tidy as people have led you to believe. So I think I'm nearly there. Oh, it's brilliant. I've got no time for questions, especially about the last bits. So, I mean, I'd kind of try to emphasize that, strategy, smattery, or whatever they say. So serendipity played a large part in a lot of what we did. It became systematic, sort of, in places. But so chance meeting and collaborations, unforeseen results, ideas with unforeseen explanatory powers looked clever, but actually it was just luck. And then, the process of revisiting conventional wisdom pops up repeatedly. Now, if I go back to my university and I say, well, what we were doing here, we were networking to start off with. We generated some pilot results. We generated a theory and then we iteratively tested it. We're all complicit in that lie with universities. And that's why, we'll find our privileges, so-called, to have our own say in what we do taken away gradually because they think it's all formulaic, and it's just not. In many cases, there are bits and pieces that come out as a bit formulaic, but as I say, if we keep telling them that the reason we're sort of successful or not is based on strategy, then we're doing ourselves a disfavor, I think. And you probably have the same sort of awful. I said it again. Sorry, we have to cut this bit. awful surveys that come round and say, so what would make your life better? What would make your life better? Or if we had a nicer canteen? No, if we have more time. And we don't mean more time to execute our well worked out plan of strategy for our research over the next 5 years, because to be honest with you, I usually can only go one experiment in advance because it would go wrong at some point, but also because that's my limited imagination. So I think it's important that we try not to go along with this emphasis on strategy and try to salvage some academic freedom to do silly things, and that's what I've done. Now this is because the committee, they're terrible. They make the honorary president do silly things. So this is me saying thank you to the people that are not here, and me saying thank you to you for listening. And now I'm going to bow.